In the early decades of the 20th century, poliomyelitis, commonly known as polio, was one of the most feared diseases in the world. It struck suddenly, spread rapidly, and mainly affected children. What made polio especially terrifying was its unpredictability. A child could appear healthy in the morning, and by evening lose the ability to walk, breathe, or even survive. The virus attacked the nervous system, leading to irreversible paralysis in many cases. Hospitals filled with patients dependent on iron lungs, large mechanical devices that helped paralyzed individuals breathe. Families lived in constant fear, particularly during summer months when outbreaks were most common. Polio was not limited to poor or underdeveloped regions. Major outbreaks occurred in the United States, Europe, and other industrialized nations. In the United States alone, tens of thousands of cases were reported annually during peak years. Schools closed, swimming pools were shut down, and public gatherings were restricted. Despite advances in medicine, there was no effective cure for polio. Treatment focused only on managing symptoms and preventing complications. This global health emergency created an urgent need for a reliable and scalable preventive solution. By the mid-20th century, scientists had made progress in understanding the poliovirus. This led to the development of injectable vaccines based on inactivated, killed virus. These vaccines played an important role in reducing polio cases in countries with strong healthcare systems. However, they also revealed significant limitations. Injectable vaccines required trained medical personnel, sterile needles, and controlled clinical environments. These requirements made mass vaccination campaigns difficult, especially in low-income and remote regions. Another challenge was cost. Producing, transporting, and administering injectable vaccines at scale demanded resources that many countries simply did not have. In addition, injectable vaccines primarily protected individuals from developing severe disease, but were less effective at stopping the virus from circulating in communities. This meant that even vaccinated populations could still contribute to transmission, making global eradication difficult. Public health experts began to realize that while injectable vaccines were valuable, they were not sufficient on their own to eliminate polio worldwide. The idea of an oral polio vaccine addressed many of these challenges directly. An oral vaccine could be administered easily, often as a few drops on the tongue, without needles, syringes, or specialized medical training. This simplicity made it ideal for mass immunization campaigns, particularly in regions with limited healthcare infrastructure. Community workers could vaccinate thousands of children in a single day, even in rural or densely populated urban areas. From a biological perspective, an oral vaccine offered another major advantage. Because polio enters the body through the digestive system, an oral vaccine could stimulate immunity directly in the intestines, where the virus first multiplies. This type of immunity not only protected the vaccinated individual, but also reduced the spread of the virus within the community. In practical terms, this meant that widespread use of an oral vaccine could interrupt transmission chains and protect even those who were not vaccinated. For global public health, this characteristic was critical. When the oral polio vaccine was finally introduced and adopted internationally, its impact was immediate and measurable. Large-scale vaccination campaigns led to dramatic declines in polio cases across multiple continents. Countries that had experienced annual epidemics saw case numbers drop within a few years. The vaccine proved to be effective, affordable, and adaptable to diverse public health settings. It became a cornerstone of global immunization programs and later of the worldwide effort to eradicate polio entirely. One of the most remarkable aspects of this success was the philosophy behind it. Albert Bruce Sabin chose not to patent the oral polio vaccine. By doing so, he ensured that it could be produced widely and distributed at low cost, without legal or financial barriers. This decision reflected a commitment to public health over personal profit and allowed millions of children to be protected regardless of their country's economic status. The success of the oral polio vaccine transformed polio from a global terror into a preventable disease and laid the foundation for one of the most ambitious disease eradication efforts in human history. Albert Bruce Sabin was born on August 26, 1906, in Bialystok, a city that was then part of the Russian Empire and is now located in Poland. He was born into a Jewish family at a time when political instability, economic hardship, and social discrimination were common in Eastern Europe. These conditions influenced many families, including the Sabins, to seek better opportunities elsewhere. When Albert was still a child, his family immigrated to the United States, settling in New York City. This move would prove decisive for his future, as it placed him in an environment where education and scientific inquiry were more accessible. Growing up in New York, Sabin was exposed early to the realities of immigrant life, hard work, adaptation, and the need to prove oneself. His parents emphasized education as the primary path to stability and success. From a young age, Sabin showed strong academic abilities and a particular interest in how things worked, especially in the natural sciences. Living in a city with advanced medical institutions and universities also meant that he was surrounded by examples of scientific progress, which helped shape his early ambitions. Sabin's formal education reflected both his intellectual ability and his disciplined approach to learning. He enrolled at New York University, where he pursued studies in medicine. During his medical education, he demonstrated a strong interest in research rather than clinical practice alone. He was particularly drawn to microbiology and virology, fields that were still developing, but held great promise for understanding infectious diseases. After earning his medical degree, Sabin continued his training through internships and research appointments that allowed him to deepen his expertise in viral diseases. He worked with leading scientists of the time and gained hands-on experience in laboratory-based investigation. These years were crucial in shaping his scientific mindset. Sabin learned to value controlled experimentation, careful observation, and skepticism toward untested assumptions. This foundation later enabled him to challenge prevailing ideas about vaccine development and pursue alternative approaches based on evidence rather than consensus. As Sabin advanced in his career, he established himself as a serious and methodical researcher. He worked in several prominent research institutions where he focused on viruses that affected the nervous system. His work was marked by patience and precision, qualities that were essential in an era when laboratory tools were far less advanced than they are today. Sabin believed that meaningful scientific progress required long-term commitment rather than quick results. One defining aspect of Sabin's professional character was his independence of thought. He did not hesitate to question dominant theories if experimental evidence suggested alternative explanations. At the same time, he maintained a strong sense of responsibility toward public health. He viewed medical research not as an abstract intellectual pursuit, but as a practical means to reduce human suffering. This perspective guided his choices and sustained his motivation during years of slow and demanding research. Despite his intense dedication to science, Sabin also maintained a personal life that provided stability and support. He married and had children, forming a family that accompanied him through various stages of his professional journey. While detailed records of his private life are limited, it is clear that his family played an important role in his life, offering continuity and grounding amid the pressures of scientific work. Balancing family responsibilities with an ambitious research career was not easy, particularly given the long hours and mental focus required for laboratory research. However, Sabin managed this balance by maintaining a structured and disciplined lifestyle. His personal values, commitment, responsibility, and service were consistent across both his professional and private life. These qualities helped him sustain the long-term effort required to achieve breakthroughs that would later benefit millions around the world. By the 1930s and 1940s, Albert Bruce Sabin had firmly established himself as a virologist with a strong interest in how viruses interacted with the human immune system. During this period, polio remained one of the most urgent medical challenges, and many researchers were attempting to understand its behavior inside the body. Sabin focused on studying how the poliovirus entered the human system, how it multiplied, and how immunity could be developed against it. His laboratory work emphasized careful observation and repeated experimentation, which allowed him to identify patterns that were not always obvious to others. One of Sabin's key scientific conclusions was that polio infection typically began in the digestive tract before reaching the nervous system. This insight was critical because it suggested that immunity needed to be strong at the intestinal level to prevent both illness and transmission. At the time, this idea was not universally accepted. Many researchers focused primarily on preventing paralysis rather than stopping the virus from spreading. Sabin's insistence on understanding the full infection process set the foundation for his later work on oral vaccination. As vaccine research progressed, two main approaches emerged: vaccines using inactivated virus and those using live but weakened or attenuated virus. Sabin became convinced that a live attenuated vaccine offered advantages that could not be achieved with killed virus formulations. Based on his research, he believed that only a live virus could stimulate strong and lasting intestinal immunity. This position placed him at odds with some prevailing scientific opinions, but Sabin remained focused on experimental evidence rather than popularity. Developing a live vaccine posed significant challenges. The virus had to be weakened enough to be safe, while remaining strong enough to trigger immunity. Sabin spent years testing different virus strains, passing them through laboratory cultures and animal models to reduce their ability to cause disease. This process was slow and required extreme caution. Any error could result in a vaccine that was either ineffective or unsafe. Sabin's persistence during this period demonstrated his commitment to scientific rigor and public safety. Once Sabin had developed vaccine strains that appeared safe and effective in laboratory studies, the next step was large-scale human testing. At the time, conducting such trials in the United States was difficult due to regulatory, logistical, and political constraints. As a result, Sabin collaborated with international partners, including public health authorities in the Soviet Union and other countries. These collaborations allowed for extensive field trials involving millions of participants. The results of these trials were carefully documented and independently reviewed. Data showed that the oral vaccine not only protected individuals from polio, but also significantly reduced virus transmission within communities. This evidence was critical in convincing the global scientific community of the vaccine's value. The success of these trials highlighted the importance of international cooperation in addressing global health challenges and demonstrated that scientific progress could transcend political boundaries. Following the success of the international trials, the oral polio vaccine gained widespread acceptance by health authorities around the world. It was adopted into national immunization programs and became a central tool in global polio control efforts. Sabin's work was widely recognized within the scientific community, and he received numerous honors for his contributions to medicine and public health. Perhaps the most enduring aspect of this phase of Sabin's journey was his ethical decision regarding the vaccine's ownership. Sabin chose not to patent the oral polio vaccine, stating that it belonged to the people. This decision ensured that the vaccine could be manufactured widely and distributed at low cost, making it accessible to countries regardless of economic status. This choice reinforced Sabin's belief that scientific discoveries, especially those with profound public health implications, should serve humanity first. The oral polio vaccine, commonly known as OPV, is a vaccine made from live but weakened strains of the poliovirus. These strains are carefully selected and modified so that they can no longer cause disease in healthy individuals. The goal is to expose the immune system to a harmless version of the virus, allowing it to recognize and respond effectively to future infections. Unlike injectable vaccines, OPV is administered orally, usually as a few drops placed on the tongue. This method of delivery is simple, painless, and well-suited for large-scale immunization programs. OPV contains 3 different attenuated virus types, corresponding to the 3 known types of poliovirus. This ensures broad protection. Once ingested, the weakened virus passes through the digestive system, where it begins to replicate at very low levels. This limited replication is enough to stimulate the immune system without causing illness. The design of OPV reflects a deep understanding of both virology and practical public health needs, making it especially effective in preventing the spread of polio. The effectiveness of OPV lies in how it activates the body's immune defenses. When the weakened virus replicates in the intestines, it triggers the production of antibodies, particularly immunoglobulin A, IgA, in the gut lining. These antibodies play a crucial role in blocking the poliovirus at its point of entry. As a result, vaccinated individuals are protected not only from developing paralysis, but also from carrying and transmitting the virus to others. In addition to intestinal immunity, OPV also stimulates systemic immunity by prompting the production of antibodies in the bloodstream. This dual protection is one of the vaccine's greatest strengths. It means that even if the virus enters the body, it is neutralized quickly and efficiently. Over time, repeated vaccination strengthens this immune response, creating long-lasting protection at both the individual and community levels. One of the most important public health advantages of OPV is its ability to reduce and eventually interrupt poliovirus transmission. Because the vaccine virus replicates in the gut, it can be excreted and spread to close contacts in environments with poor sanitation. While this might sound concerning, it actually provides an additional layer of protection. Contacts may be indirectly immunized through exposure to the weakened virus, a phenomenon known as secondary spread. This effect helps build community-wide immunity, especially in densely populated areas. This characteristic made OPV particularly valuable in regions where polio was endemic. Large-scale vaccination campaigns using OPV were able to rapidly reduce the circulation of wild poliovirus. As fewer people carried the virus, outbreaks became less frequent and eventually disappeared in many regions. This capacity to stop transmission is why OPV became a central tool in global polio eradication initiatives. Although OPV has been highly successful, it is not without limitations. In rare cases, the weakened virus can mutate and regain the ability to cause paralysis, leading to vaccine-derived poliovirus cases. These events are extremely uncommon, but they have influenced modern vaccination strategies. As polio cases declined worldwide, public health authorities began transitioning from OPV to inactivated polio vaccines in some regions to eliminate even this small risk. Despite these challenges, OPV's contribution to global health is undeniable. It has protected billions of children and reduced polio cases by more than 99% worldwide. The vaccine's technical design, ease of use, and ability to interrupt transmission made it indispensable in the fight against polio. Albert Bruce Sabin's scientific approach and ethical commitment ensured that this technology served humanity on a global scale, bringing the world closer than ever to the complete eradication